much. Thank you very much for the invitation. Thank you. So you'll see we are going to be doing some practical work, so I will be requiring volunteers. But to get going, we're going to see how your zoological knowledge is doing. Or we're going to skip over who this is, fairly obviously, but I want you to shout out, what's this? This is a nose. What animal is this? Very good. No, this is a mole. So this is called a star-nosed mole. Now, the school kids I do this with all know the answer because it's in GCSE, but those of us who are slightly older don't know. So this is a mole, and in fact, these are both touch and taste and smell sensitive papillae on the outside of its nose, and those are its nostrils. So yes, this is an eel. What kind? Well, it's either a moray or a conger. I can't remember. That's a bat, fairly obviously. Elephant, no problem, right? Cat? Dog? Okay, now we're getting on to the difficult ones. Those are all easy. Right. This is a nose. It's not a fern. Sorry? What do you think it is? Well, it's an insect. Okay, so well, you've failed on that. In the old days, medics used to have to know about botany and entomology. That's obviously all gone. This is a moth antenna, and in fact, it's from a male moth, and we'll see an example of that later on. Now, this one. No, well, close. It's a bee. This is a bee antenna. This. No, sorry, this. I'm not looking. This. What is it? It's a beetle, and it's a cockchafer, and this is a male cockchafer's antenna, and we'll come back to that later on. So the point about the sense of smell is just to give you some idea of the range of different structures there are in the natural world that have evolved to detect odours. And the sense of smell, which is basically simply the detection of molecules not by contact, that would be taste, the diffusion of molecules in the sea or in the air, that is basically the sense of smell. And it has been around for billions of years, in that even bacteria are able to detect chemosensory gradients and to move up and down them. Now, why would you want to smell? Well, there are lots of reasons. Fairly obviously, you might want to smell to be able to find food. Particularly interesting thing is to memorize things. So for me, and maybe this is the same for you, Ian, hot Vimto and cheese and onion crisps and the smell of Stockport baths and the sound of the echoey, that smell of the chlorine and the echo of the old baths, they all go together because we go to Stockport baths, maybe Ian was there, and then afterwards I'd have hot Vimto and a packet of Golden Wonder cheese and onion crisps. It took me ages to find that ancient 1960s packet. So what happens in virtually any animal which has got a brain, any organism that's got a brain, when it detects a smell, it does two things. It puts part of that to working out exactly what the smell is, and then the other part of that signal, so it decides what it wants to do. Does it like the smell or not? The other part gets put into memory. So anything that's going on at the time tends to get associated with the smells and the tastes that you're experiencing. So you can get very, very powerful memories called single trial learning, whereby if you are sick, then the smell and the taste of a particular food that you were eating when you were sick, they're going to be associated for a very, very long time. For me, it was 30 years before I could smell cauliflower without wanting to vomit. So this is a very, very powerful system that works in all organisms. You might also want to find a mate by using volatile signals. So here we've got two crane flies. Sadly, we don't get them quite as beautiful as this in the UK. And this is the male. And you can see he's got these very, very long antennae, very, very feathery, a bit like we saw in the moth. And this is the female who's got much shorter antennae. She only needs to be able to find a place to lay her eggs. He has to find her. And that can be very, very difficult if she's a long way away. So the substances, that he's detecting are called pheromones, and these are chemical signals which are given out by one organism which alter the behavior of another. And here's the male moth. This is close on. You can see his eyes here. So we're looking at him head on. And he's got these absolutely enormous antennae. You can see these loops. And on the loops are thousands of hairs. And on the hairs are millions of receptors that mean that he can detect a female from miles and miles away. He's got an incredibly sensitive sense of smell. I'm going to show you quite how quick this can work in a minute with this little film. So what we've got here are some very, very fat silkworm moths. So silkworms have been used for about 3,000 or 4,000 years to, in fact, I saw a paper today that they found silk in a tomb from 6,500 years ago. So for a very long time, we've been rearing silkworms and extracting the silk from the pupa, which is where the silk comes from. So silkworms now are pretty porky, and you're going to see these chaps here can't fly. We've got a female here who's under a little lid, and this is a fan. And when I turn the video on, a hand's going to appear, lift up the little dish, and the smells will blow, and we'll see the males doing their best to get to the female. Here comes the hand, lift it up, and instantly the males start to flutter their wings. We'll zoom in on. They can't fly. They really are very, very overweight. They will need to be on a bit of a diet. So they're charging off to try and find her. This one's hopeless. So who's going to get there first? She's diffusing her pheromones. He's won. He's lost. That's the race of life. So he's responding simply to a chemical compound produced by the female, which enables him to identify her sex, her species, and then to locate her. So those are all sorts of reasons why you might want to be able to smell, but the real reason you need to be able to smell, and this is particularly significant for clinicians, is you need to smell in order to be able to taste, which might seem kind of counterintuitive, but I'm going to demonstrate that to you, because you taste with your nose. So we're going to do a little experiment. So what I want you to do is, these are jelly beans. Take one and pass one along. You have to, if you can take one and pass one along. Don't eat it yet. Don't sniff it. If you can take one, if you can take one, pass it along. So I want you all to take a jelly bean. These, oops, I dropped some. These are vegan, halal, whatever. That's very important. You know, I do this in schools, and it's very important for the kids to know. If you don't want to do it, you don't have to. It's fine. But there is no animals of any kind have been harmed in the making of these. So you're going to take your jelly bean. Don't eat it. Don't taste it. Okay. Right. Nobody got one? Everybody wants to do one? Got one? Right.
Hold your nose. Hold your nose. Get your jelly bean. Put it in your mouth. Oh, just chew. Keep your fingers on your nose.
Tastes vaguely sweet, maybe. Take your fingers off your nose. Now you know what flavour it was. Probably Vimto if you're lucky. If not, it was grape. But you can see the difference between the moment when you had just had your senses of taste. I've got to get it out of my mouth now. Just had your sense of taste. It was, your sensory experience was pretty crude and not at all rich. As soon as you can smell, then you get the whole flavour experience. And that's why when people lose their sense of smell, it can have such a devastating effect on their quality of life, because if you can't smell, then you are limited to the number of dimensions, probably about 5 dimensions, 6 maybe, of taste, whereas the dimensionality of smell, I'll explain what that means later on, is potentially infinite. Now, you all know, or you knew once upon a time, how it works. Because you're not smelling, your olfactory neurons aren't down here, they're up here, in fact, roundabout at the level of your eye, which ordinary members of the public find quite remarkable. So you're smelling, what you've just done, that experience you've just had, has come about because we've built up pressure here, we've been blocking off your nose here, we've built up pressure here, you take your fingers off your nose and then whoosh, all that air goes swooping past these neurons which are in your olfactory epithelium, which are dangling down, bits of your brain dangling down through tiny little holes in your cribriform plate, if you can remember doing that dissection. So that is how we're smelling. We're smelling with this part of our nose, and that gives us that whole sensory experience of taste. So you taste with your nose and you smell with your brain. Those are 2 important things that we need to know. So, which of these has got a better sense of smell? Dog. Everybody says dog. Well, it's not quite so simple as that. So a dog has got far more sensory neurons than we have. We have about 4 million. A dog has probably got about 8 million, and we have about 400 types of olfactory sensory neuron, and the dog has maybe about 500-600. But above all, what a dog is better at, rather than detecting a range of odours, is the threshold is much, much lower. So dogs that are able to sniff around, you can see them following stuff which we can't smell. It's simply because, in general, their threshold is much, much lower. So, question now comes up, what can we smell? So I need 4 volunteers, please, to come up to the front and stand here and try and... I'll pick on people if you don't come. So just 4 people. Come on down. Come down to the front. You've got to stand here. You're not allowed to say, yes, sir, excellent. 2 more people, please. 2 more people. Come on, sir, you at the front. OK, stand here at the front. And you've got to stand here. Oh, Ian's doing it as well. Fantastic. 5 of you. Brilliant. That's fine. 5's good. 5's good. 5's good. 5's good. Now, you're not allowed to look behind you because I'm going to show people what the various smells are. So we're just going to be doing some simple smells to start with. It doesn't really smell. What if I put it on my hand? Warm it up like those orange ladies you get in the fancy shops. OK. How's that? If you smell it, a bit of warmth. Don't say what it is. So this is just something very, very straightforward. The audience can see. OK. Any idea? Any idea what kind of thing?
That's citrus.
Citrus, very good. Well, that's orange, so that's pretty good. That's pretty good. OK, right. This one, a bit more difficult. OK, well, I think we've probably got the wrong profession here. OK. Something like wood. Cinnamon, cloves, very good. Right, now I say cloves, you go, ah, yes. So it's very interesting. You always have this ha-ha tip of the tongue phenomenon. Right, this one is in a box. Only once has anybody ever got this right, literally right. So it's a bit smelly. Any ideas? Odd mothballs. Manure, right, very good. But what kind of manure? Any? This is what somebody got absolutely right. Pig? No, turn around. You've all been trampled to death. You're all been trampled to death by an elephant. OK, so we can see that there's various, once you, well, maybe not with the elephant, but with the everyday smells, once you can recognize it, then it kind of clicks into your head. But I want to prove to you that, in fact, you've got a much better sense of smell than you think. I want to prove to you that you've got an atomic nose, that you can smell atomic differences. OK, so I'm going to take this odor first. Which one is this? P or C? This is C. OK, we're going to smell this. OK. So this is used in cooking. Cumin, close. Begins with C. It's close to cumin. Caraway. It's caraway. OK, right. Now this one. The audience might have noticed something. This is much more. This you should be smelling a couple of times a day, I would hope.
Can I ask,
No.
picture you're showing on the screen, is that the whole substance?
Yeah. Any ideas? Toothpaste, peppermint, spearmint, right. But if you turn round, everybody, what can you see? So they're optical isomers. So they have exactly the same structure, but they are simply, they're like 2 fingers, 2 gloves with exactly the same structure, exactly the same composition, but they're mirror images of each other. And once you know that, you can actually start to smell bits of kind of spearmint, peppermint in the caraway smell. You can just start to get the hint of it, but they produce qualitatively different experiences to us. OK, right, now I'm going to show you something you won't know, well, because you're scientists, maybe once I did have somebody who was able to identify these because he worked in a lab. So I'm going to give you 2 smells now that you won't be able to identify, but I want to know, can you detect a difference between them? There's a very subtle difference, OK? You won't be able to identify them. Yes, some people can find it quite. Yeah? It's not massive. In fact, it's very hard to get smaller. Yeah. That one. And that one. And that one. Could anybody detect a difference there? Yeah? Okay, right. Well, what you have just detected is a single atom of carbon. Those two, look, turn around, there are heptanol and octanol, which differ therefore by a single atom of carbon. People say that heptanol is violet, sweet, woody, and octanol, which is in fact what they put in orange-smelling washing up liquid, smells sweet orange rose. And that single atom of carbon produces a very subtle difference that when you've got the two smells together, you can detect the difference. So, right, thank you very much. You can give everybody a round of applause. That's great. We'll be doing some more later on. Okay, so this is suggested you've got an incredibly powerful nose. So how many odours do you think we can smell? Throw out some numbers. 700, okay. Higher or lower? Higher, right. Higher, and we don't want to be here all night, so give us another number. 10,000, higher or lower? Higher, give us another number. Million now, we're starting to talk. Higher or lower? This is your last chance. Okay, you haven't been paying attention to the scientific literature, obviously, so there's a big argument about this. We don't know, but it's conceivably, there is an argument that it's over 1 trillion odours. So, in fact, I mean, basically it's infinite. We do not know what the limit to the human nose's ability to be able to detect odours is. And just to give you some ideas of quite how rich our sensory experience is, I'm going to explain how this might work in a minute. But if you just think about ordinary smells like camembert, this is just some of the different compounds that are going to the smell of camembert. There's a whole set, there's far more, but these 4 are the primary ones. So any particular odour that we will smell every day is composed of different blends in different concentrations producing that sensory experience. A rose, for example, if you extract the molecules, the volatile organic molecules from a rose scent, there are about 250 different molecules involved. Now, that doesn't mean to say when you smell a rose you're getting all 250, but the potential is there for this immensely rich sensory system. So, how does it work? That's the key question I'm going to be dealing with in the next period. How does this sense of smell work? Well, as in most things, if you want to have any idea about anything, you have to ask the ancient Greeks. And this chap here, Democritus, he basically came up with the theory of atoms. He came up with a pretty good idea. He's sitting there under his olive tree in Greece in 4th century, 5th century BC, swilling his retsina, just thinking about stuff. And he decides that the whole of the universe, everything which looks solid, is in fact made up of these tiny particles that you can't see, which he calls atoms. So he's the beginning of the whole atomist philosophy. And he not only thinks about the retsina and the tree and his head and all the rest of it, he also starts to think about sensory experience. So he decides that probably things that smell nice, the atoms must be kind of round, and things that smell horrible and acrid must be kind of pointy because they get stuck in your nose. And that's basically it, to be honest. We don't actually know what the link is, but there's a link of some kind between the molecular structures we saw with the caraway and the spearmint and our sensory experience. The big breakthrough in how it might work came somewhat later, in 1991, where these two people, Linda Buck and Richard Axel, isolated the genes that they suspected encoded the olfactory receptors in rats. Turned out they were right, and they got the Nobel Prize for this in 2004. So again, you might remember this or you've forgotten it. Let's just brief recap. So here we are, a close-up of that your olfactory sensory neurons going down through your cribriform plate, dangling here into the mucus, and of course your odours that we're smelling are all in the air, but your sensory neurons aren't actually in contact with the air. You don't actually smell in the air because remember, you're basically a fish. Just you've got the sea inside you, you've got the sea outside you as well. So you've got this layer of mucus on your sensory, over your sensory neurons, which includes lots of odor binding proteins which enable the volatile hydrophobic molecules to get through this layer of mucus, which protects the neurons, and then to bind to the receptor. So each receptor neuron, each type, they're color-coded here, expresses just one of your 800 alleles. You've got about 400 receptor genes, and you've got 2 alleles, obviously. So you've got 800 possibilities. Each neuron can decide to be one of 800 things. Quite how it decides which of the 800 it's going to be, we don't know. There's still a lot of argument about this. But each neuron expresses just one type of receptor, and all the neurons expressing the same kind of receptor, here red, go to the same place in the brain, in the olfactory bulb. This is called a glomerulus, and it's the first staging post of sensory processing. Now, okay, that's fine. But we've got 400 olfactory receptor types. So how can we get from 400 to, well, even 1 million, never mind 1 trillion? How can we get to such a complex sensory processing system? Well, this is what we think happens. Each neuron, each kind of neuron can detect more than one kind of odor, and each odor can stimulate more than one kind of neuron. So here we've got a little model. We got these odors. Here are the 2 odors we were looking at, heptanol and octanol, and you can see they're just differing by how many carbons they've got in the chain. And in this model, we've got our 14 receptor types. That should really go on down up there, way into the center of the hospital, because we've got 400 receptor types. But in this little model, heptanol activates receptor 2 and 5 and 6, and octanol doesn't activate 2, but it does activate 4. It also activates 5 and 7 and 10. So you're getting mixtures, you're getting slight changes in the profile of your sensory neuron response, which enables you, by this combinatorial code, to effectively detect an infinite number of odors. Now, how does it really work? That's a little model. And then the 3 most important words in science, which I always bore my students with, are, we don't know. So that's the really short, so we can go home now. We don't know how it works. We don't know what the relationship is between each particular molecule that is going to be a smell and the structure of our olfactory receptors. You'd think that it would be easy to put it all into a computer to say this is what the protein looks like, this is how it should detect the odor. It doesn't work that way. We can't do that. We haven't been able to crystallize any of these receptors yet, so we don't know even what structure they actually have, what their 3-dimensional structure is. And above all, we don't even know what we're actually detecting. Are we detecting the smell, that heptanol, or are we detecting heptanol and the odorant binding protein, which is kind of a chaperone, which takes the smell through that mucus layer and puts it onto the receptor? So this is all a bit of a mystery still. Most people think that it is actually the smell we're detecting, but how it binds, how it actually works, we don't know. So the question then comes is, how on earth can we find out? Well, when I began working on this, there were various options that were put to me about how we could work on the sense of smell. This was one of them. I didn't particularly like this idea, so instead I chose this, which is the maggot of the title. So these are Drosophila maggots in their natural environment, which is a piece of rotting fruit. And this is that little one that was down on the corner. This is close up and personal with a Drosophila maggot. And these things here, marked D, are not its eyes. These are its noses. They're little domes. The eyes of a maggot are basically patches of about 15 neurons deep in its body, and all it can really do is tell light and shade. Its sense of smell, however, is really quite acute. So just to give you some idea about why I chose the maggot rather than sniffing men's armpits, you've got 4 million smell cells into 400 cell types. A maggot has got just 21. And each of those smell cells is unique and different, expresses a different olfactory receptor neuron. So here we are. This is the smell cell here. The T thing is where it's doing its tasting. And if you go zoom in on this dome, you can see it's got these slight pore structures in it, and through these pores that the smell goes in. And this is a nice diagram done a long time ago, but it shows you that those 21 neurons are in 7 bundles of 3, and the dome is basically the dendritic arborization of those sensory neurons. They branch and branch and branch. The smell comes through here, activates these neurons, which then fire, and then the maggot decides what it wants to do. Now, these are not your average fishing maggots, if you've probably already guessed. These are the maggots of Drosophila melanogaster, which you may remember. This is the tiny little fruit fly, which, if you have forgotten, you won't have forgotten last summer. You'll have had them floating around in your fruit bowl, or if you drink alcohol, if you go outside, you'll get them hovering around your alcohol. And this is, for those of you who want to know the Greek and Latin origin, it means the dew lover with a dark abdomen. Here's the dark abdomen. This is a male. And you can tell he's a male because he's got the dark abdomen here, and he's got these things here, which are called sex combs, which he uses during mating. And here is antennae with these little feathery bits, which are mainly used to detect sound. And where he's actually doing his smelling is on this kind of big round thing at the front. So this has been, as you know, Drosophila has been used by geneticists for over 100 years. It is incredibly powerful. I'm going to show you kind of some of the stuff we can do with it. The downside of it is you may have forgotten how small they are. So this is a USB key, and that's the fly. And the maggot is a bit smaller than an adult fly. So it is very, very small. So doing the kind of experiments I'm going to be describing in a minute to you is really pretty tricky. It's very fiddly. But the great thing about these things, and I initially worked on flies, and I was studying pheromones in flies. I'm very interested in that. And the person I was working with in France said, why don't you try using maggots? And my response was, I don't want to work with them. They're boring, which was the wrong answer, because that's the reason I did want to study them, because they are so boring. Flies, you might think they're kind of stupid, but if you try doing a complicated experiment with a fly, it's not interested, it's got a headache, it doesn't want to mate, it doesn't want to respond to the smell. It can move in 3 dimensions, you know, the clue's in the name. A maggot is very, very stupid. They're highly genetically manipulable, like their adult form. They're always hungry, so, and this is basically all they do, they just mooch along. They are very simple, and they are incredibly stupid. They're not interested in sex. They can't fly. They're not really interested in light. They're just interested, like the hungry caterpillar, getting very, very big and then turning into an adult. So this is basically what a maggot does. It mooches forward. It can put its head from side to side. If it gets very, very excited, it'll rear up. And this is an example. I'm going to show you a quick little video. We've got some quite old maggots here, a bit older than what I normally use. We've got a smell here, no smell here. Speed it up so it's a bit Benny Hill, but there's no Benny Hill music. You can see that as soon as I put them in the middle, they start moving. They're moving randomly at first. Some of them are a bit stuck together. But very, very quickly, you end up with more on the side that there's a smell than you do on the other side. They start to move towards it. Some of them are a bit dim. They haven't actually smelt the smell as it's gradually diffusing. So there's an odor gradient building here, and this smell they like, and they're moving towards it. So it's very straightforward to ask a maggot, did you like that smell, or didn't you? You can count the number of maggots on this side, number on that side. You get a nice little index of quite how much they like it. But I'm not primarily going to talk about behavior. I'm going to talk about something much more interesting. Because this is Drosophila, we can make a maggot with a single smell cell in its nose. So here we've got a maggot. This is the little dome thing, and we're expressing GFP, green fluorescent protein, in each of those 21 cells here. Here we've got a maggot with just one cell has got GFP in it. And in fact, we can make that maggot so that only that cell will work. So we can record electrophysiologically the actual responses of a single cell and see what does it look like, what information does it send to the brain, how does it respond to all those amazing odors, what exactly is it doing. And we can also say, well, if you're a maggot with just one smell cell, how do you respond? What's your behavior like? So we can integrate the whole thing. This gives you an idea of the very high-tech system we have. We have a matchstick, and on the matchstick is the maggot. The matchstick's nice and damp, so the poor maggot's going to stay alive. It's been wrapped round with parafilm, and then the poor old thing's got one electrode shoved into its body here, and then a very, very thin, you can see these little 2 dark patches, those hook things we saw on the big scanning electron microscope image. So the maggot's lying on its back. It's been squeezed so that its eyes or its domes are bugging out, and we can then put the electrode down by the side, record from it, blow air with smells over it, and then see what happens. And I'm just going to give you an idea about what a single maggot cells can do. And this will tell you something very important about the brain, how the brain works. Because people often say the brain's like a computer. It's not. It's an interesting analogy, but brains are not computers. Even the simplest neuron is not working on a binary basis. So here we've got 2 kinds of cell. These are actual real data. we've got very exciting names, 24A and 74A. So these are cells expressing the 24A receptor or the 74A receptor. And we blow a series of odors over. This is what the little bar here is. And you can see this cell doesn't particularly care for butanol, which is an alcohol with 4 carbons in it. 74A, on the other hand, gets very, very excited and fires very, very strongly. OK, so it's got a nice qualitative difference. Octanol, on the other hand, with this cell, actually turns it off. This particular cell was firing very strongly, just randomly, spontaneously firing. When we put the smell over it, it actually inhibits it. This cell actually gets very excited again, although it doesn't show quite the same response. But it's giving me a very, very strong response. So the same odor can activate or inhibit. So it's not just 0 or 1. This is -1. It's actually inhibiting the cell. And that's information the brain can detect. Or you can do something more complicated. This odor activates both neurons. This neuron, it activates it very, very strongly. And then when the odor stops, the neuron just shuts off. This neuron gives us this double hump. It fires, and then it fired again. So each smell cell combination is producing a unique response. You can actually tell by looking at the recording what smell combination you're looking at. And the same thing is happening in your noses right now. It's incredibly rich. And basically what you're getting is the brain is noticing any change from spontaneous activity. And that can be activation or inhibition, or it might be no change, which is also information. So this, if the brain wants to know, am I smelling butanol, it needs this activated very strongly, but it also needs 24A to say, I didn't smell anything at all. That's part of the whole code that the brain is interpreting. So we've got more, less activity, and then differences in the intensity of the response. You can see here, each of these cells, this cell is responding in different ways to these 3 odors. But you've also got a change over time. There's something, it's not simply an on-off response. We've not got one number here. We've got a change over time as well. And this is something we've got very interested in. So this is a bit of a challenge. We want to be able to prove, this is something we've just got a grant into the BBSRC for at the moment. We want to be able to prove that the timing of the activity of the cell, not simply is it activated or does it is it inhibited, but the shape of its signal, that might be informative for the brain. How can we do that? Well, the answer is we're going to make a maggot smell blue, which sounds a bit surreal, but in fact this is using a very recent technique which you may have heard of, optogenetics. So basically we can transform those smell cells into an eye. We can make it detect light and see how that responds. So we put a photoreceptor molecule into that smell cell. And we can then manipulate light in a way we can't manipulate smell. It's very, very difficult to manipulate smells, to turn it on and off and give it in precise pulses. With vision, it's dead easy. So we can, in fact, convince the maggot that it's smelling something by activating its neurons, its smell neurons, using light. When I told my daughter about this, she said, but Daddy, that's maggot mind control. It is. Okay? So how the maggot reacts can tell us something about the importance of different structures of the signal going to the nose. So here's to give you an example. This is a single maggot, and this is on a small plate, and there's no smells involved in this at all. You're going to see a few seconds of it just mooching around. The computers mark the head and the tail and where the head and the tail are going. Then you're going to see 2 flashes of light. And you'll see the significance of the 2 flashes of light in a minute. And then you'll see what the maggot does. And a normal maggot without this special receptor in it doesn't do anything, doesn't care about the blue light. 2 flashes of blue light. And then, OK, computer, this is what it was doing beforehand. Now it's thinking about it. And it's going off in a straight line. It thinks it's just got a whiff of something nice, and it's moved off in that direction. So we've created the illusion of a smell in this maggot's nose. That's quite fun, but how are we going to use that to get at the idea of timing? OK, so here's some real data. We've got if a 200 millisecond pulse of blue light here, just like we saw, 2 flashes of blue light, bang, bang. And each of these pulses of blue light, when we record from it, produces about 8 spikes. The neuron sends 8 bits of information down the wire up into the brain. Slightly fewer in the second one, you'll notice, each time. So it's about 8 and 7. So you end up with about 14 bits of information produced by those 200 milliseconds. But we can separate those 2 pulses of light by different intervals. So we're getting the same numbers of bits of information, the same number of spikes going to the brain, but they're now distributed differently over time. So any difference in behavior with different signals shows that they're not simply counting the number of spikes, their brain is looking at how they're distributed over time. So this is a big issue in sensory neuroscience at the moment. A lot of neurons are known to have what's called temporal information in them, but with this, assuming we get the grant, we'll actually be able to prove that the poor old maggot brain can do something with it. Okay, now I need more volunteers. Same people, come on, same people, right. And then I'll hand this round because you'll all be very intrigued by this. Okay. Right. If you stand along here, very unlikely you're going to be able to detect this. Okay, right. This is a smell that I find quite sweet. You get very different responses. Some people think it's absolutely disgusting, so feel free to be revolted. Some people can't smell it at all. Some people think it's very nice. Okay. Can you smell it? Yeah? Is it okay?
That's right.
Okay, sweet?
Yes, okay.
Okay, right, this is bad. I only want somebody to say it's foul. Perhaps the gentleman at the end will. Not very pleasant, that's more like it. So how would you describe it then?
Slightly acrid.
Acrid, back alley, chaps, where chaps have been peeing. There you go. That's what I want to hear. Right, you.
I can't really smell it, actually.
You can't smell it. Okay, and final one.
Yeah, I think I agree with you.
It's foul. It's foul. Okay, right. Well, I'll let you smell it around. Now, thank you. Go and sit down. Have a round of applause to the volunteers. What you've just smelled, this is available in certain shops, I believe. And it attracts women fast, but it also attracts men fast. In fact, it does neither. The only thing it's going to attract is a sow. So if any of you are pig farmers, you'll know that this is androstenone, which the male boar produces, and he produces by scent glands around his ears, and this will then induce ovulation in the sow. So if you're a pig farmer, you spray this stuff up her nose, and then you surprise her with a turkey baster, and a few months later, you'll end up with some little piglets. So this is sold as a human sex pheromone. It is not. And the very fact that we've got this variation between people like Ian, who thought it was disgusting, and other people who thought it was quite sweet or couldn't smell it, that in itself, that variability tells you this is not a human pheromone. In fact, there are no known human pheromones. The only potential one there might be is perhaps there's substances produced by the nipple during breastfeeding that may perhaps be involved in helping the newborn find the nipple, but even that's debatable. Now, we've seen variability here. That's very interesting because this is one of the rare examples in humans where we know exactly how this works. How are we doing? Who found it Who found it a sweet smell? Out of the ways you put it. Who thought it was disgusting? Okay, that's quite a few of you. Excellent. I'm sorry about that. You probably won't be able to get it out of your noses now. So, I said earlier on, we don't actually know what the three-dimensional structure of these molecules, the receptor molecule is. But this is the classic way we draw it as a little snake diagram. You've got a cell membrane, you've got the N-terminal of your molecule outside, and a C-terminal inside. And there are a number of variants in the human genome. But I can tell you from your responses what your DNA is for the response to this odor, because it's controlled by a gene called OR7D4, which produces this protein, and single base changes change an arginine to a tryptophan and a threonine to a methionine, and those 2 amino acid changes on the outside and the inside of the cell produce those qualitative responses of thinking it's lovely or thinking it's foul. So if you thought it was nice, that like me, you probably have a T in this position and a C in this position. If you thought it was nasty, then you almost certainly have a C and a T in these 2 positions. So we can go from genotype to phenotype and back very, very straightforwardly. So the basic way it works is that, if you remember, I said we've all got, of course, 2 copies of our gene, each of our olfactory receptor genes, and if you have RT and WM, or both alleles with WM, then, like me, you think that it is quite pleasant. If you really, really didn't like it, you're probably homozygous. Both your alleles have R and T at each of these positions. Now, I got very interested in this with a colleague who's an anthropologist who wanted to see if there was any link between this allele, this gene and the alleles for it, and this response, very strong response you have, and food consumption. So we sequenced OR7D4 from over 2,000 indigenous peoples. So these aren't admixed populations like virtually all of us. These are cell lines from peoples all over the world. And you can see basically the blue is those people who think it's not so bad, whereas the greens and the reds are the people who really hated it. If I have people of an Afro-Caribbean origin, it's generally a pretty safe bet they're going to find it vile. Not always, but generally. So what we wonder, what's interesting is that the genetics, when we're studying this, the population genetics suggests very strongly that this mutants that because we know in general anything that you find in Africa is the ancestral form, okay? We'll see this is significant in a minute. So this strongly suggests that when we originally evolved 100,000 years ago, then we all would have not liked that smell. We would have all found it foul. And then at some point, and our data suggest about 10,000 years ago, alleles arose in this part of the world which enabled us to not care about this particular smell. Now, why would there be an advantage to not being able to smell androstenone or even thinking it's sweet? Well, if you're a pig farmer, you'll know that one of the things you have to do with your boar is to castrate them with a little elastic band. And then the balls fall off and they're quite okay. If you don't do that, then the meat will have what's called boar taint and will taste a bit like that stuff smells. So for those of you who didn't like it, imagine being given a rasher of bacon and it tasting like that. You wouldn't want to eat it. So one hypothesis, which we can't prove, is that in fact this is where the pig, domestication of the pig arose around about 7,000 years ago. So there's kind of a nice correlation in terms of the origin of this ability to eat pig meat and the fact that we've got this very widespread allele that means that we don't mind the smell of it. Now, we were busy doing this and trying to think about the significance of it, and I was out running by the Mersey, where I live in Manchester, and I just suddenly stopped dead because I thought, wait a minute, if we can go and interrogate these genes from these 2,000 people, what about some long-lost relatives? What about our cousins? I'm using cousins because, as you probably know, Neanderthals, these are some rather old images. This is often what I feel like in the morning, actually. Rather old reconstructions of Neanderthals. They left, we interbred with them, we now know. So we have got the genome of quite a few Neanderthal individuals. Those individuals who were found in very dry, cold conditions, the bones were found in those conditions, you can extract the DNA from them. We've sequenced the Neanderthal genome, and therefore we now know, one, that we interbred with them, and vice versa. So for everybody in this room, everybody who's not of an immediate Afro-Caribbean origin, around about 2.5% to 3% of your genes, on average, are Neanderthal. And more or less, we can reconstruct about 80% of the Neanderthal genome by looking, because it's not the same 2.5% in all of us, it's different 2.5%. We can reconstruct about 80% of the Neanderthal genome from modern humans. So Neanderthals, from a biological sense, were the same species because we interbred with them. We had babies. It might have been nice, it might have been horrible, we don't know. But those genes live on in us. Quite remarkable. So we could go and say, what did the Neanderthals think of this smell? Here's another image, rather cooler image of a Neanderthal. Completely gratuitous, although we do know that this is from the last outpost of the Neanderthals, which was in Gibraltar, where they finally went extinct about 35,000 years ago. We don't know why they disappeared. Did we outcompete them? Did we give them diseases? Did we kill them? Was they just kind of very, very fragmented populations. We're not sure why they disappeared. But we know that, in fact, these people are called the bird people because in their caves they find lots and lots of these crow feathers. So it's not entirely gratuitous to imagine. They also have these long, long fish bones, which they suggest in this reconstruction they were using for decoration. Having face paint on the face is possible, but we don't know. Now, there's not only Neanderthals. So you've all heard about Neanderthals. Many of you will probably know that story. I've just mentioned about about our genes. But what about this? Who's heard of the Denisovans? Hands up. Okay, right, that's interesting. So that's not much more than I get from a class of first-year undergraduates who've never heard of them either. So this is one reason why I'm doing this talk, because I'm on a bit of a mission to explain about this. So this is the Denisovans. This is it. In about 2007, in a cave called Denisova, in a region out in Siberia, a cave that's been occupied by humans and by Neanderthals, they found this tooth. Now, you may dimly recollect something about human teeth and look at that and think, my gosh, that's not human, because it isn't. It is huge. It is immense molar. It's much bigger even than a Neanderthal. So as soon as they found this tooth, they knew they had something. But what they then did was to extract the DNA from it. They found a tooth and a tiny little tarsal bone. It turned out to be from a 9-year-old girl. They had to destroy that bone to get the DNA out of it, but they were able, therefore, to find 2 individuals. And this was a complete bombshell in 2009 when they sequenced this and they published it in Nature, that there was this completely new group of humans who were not Neanderthals. We don't know what they look like, apart from this. We don't know anything about them, but they lived in this cave, and these Denisovans also interbred with us. So as humans left Africa, we bred with Neanderthals, at the very least in in Middle East, and then probably also in Northern Europe. We went, this is where Denisova Cave is situated. We came out through Africa, we moved across here, we mated with the Denisovans in these kind of regions. So a lot of the genes from people from Southeast Asia and from Australasia and from Polynesia, they have Denisovan genes in them because they mated with them as they went out. People like myself, most of whose ancestry is from Northern Europe, I don't have any because my ancestors never met them. We moved up there. So we can then ask the question, what did the Denisovans and the Neanderthals think of Androstana? So here are peoples, one of whom we don't even know what they look like. They've both been, the Denisovans went extinct, who knows, maybe 50,000 years. The Neanderthals, 35,000 years ago. And we can now say, what were they able to smell? So we can actually look into their sensory world by looking at their DNA. And this isn't terribly surprising, given that the ancestral human form was able to think this stuff was disgusting. The Neanderthals that we have had the same genes as those of you who really hated it. They really did not like it. So if we'd have been able to go back in time and waft that under their noses, they wouldn't have been very happy about it. They would have thought it smelled foul. Now, things got very exciting when we looked at the Denisovan DNA, because they had in there a genetic variant that is not present in any of the human populations we've looked at. As I said, we looked at over 2,000 people, and it's not present in the Neanderthals either. So the Denisovans, people think, came out of Africa first, then the Neanderthals, then us. So we had the possibility that maybe the Denisovans could sense things in a slightly different way. So to find out, we had to reconstruct a Denisovan nose. It's not quite as exciting as it sounds. What we had to do was to make a protein which contained this amino acid variant that we saw from the DNA sequence. It was then put into a cell line, and then we poured androstenone over it and saw how the cell responded. And this is where, well, this is science. This is the problem with science. It doesn't always work the way you want to work, because it was no different. Very disappointing. So this is why we got published in a journal called Chemical Senses and not Nature. If we'd found a different, which is absurd, But if we'd found a difference and we'd said, look, the Denisovans had a different response to both humans and Neanderthals, nature would have lapped it up. So here you can see this is the response of these cell lines to androstenone, and this is your and mine version, classic version, and this is the one from the Denisovans. No difference. So that was a bit of a letdown. But I think the principle that we can use genomes obtained from bones of people who've been dead for tens of thousands of years and understand something about their sensory world is quite extraordinary. So with my colleague Kara Hoover, who's the main person who's from Alaska, who did most of this work, we're now looking at some of the other human receptors where we got a pretty good idea of what the link between genotype and phenotype is, and we're looking into the Neanderthal and Denisovan genomes to try and see how they might have responded to other smells, agricultural smells, smells of plants, things that we know that there are human variants for. So, to summarize, most important thing, we don't know how it works. So I haven't told you how smell works because I don't know. Okay? The way the nose is interpreting smells is very complex. It's not binary. It's very complicated. You can use simple systems to understand complicated ones, which is obviously a major principle of biology and why people like me study flies and maggots. And we've got these astonishing possibilities come about over the last 5-10 years for discovering completely new species. We are pretty certain, in fact, we can now predict, in fact, from African DNA, from the DNA of people who live in Africa, that there are fossils to be found that we have not yet found because Africans have traces of interbreeding with a lineage that we have no idea what it is. So there were other human-ish, I don't know, people you could mate with, whether you wanted to, we don't know. Perhaps you did. Perhaps it was loving and glorious and very pleasant. Perhaps it was foul and violent. But whatever happened, you could mate with these individuals. And just as we have DNA from Neanderthals, some of us have DNA from Denisovans, people in Africa have DNA from completely unknown populations of human-like beings. So we can look back into the deep past and we can even understand something about their sensory world. And above all, it's what we don't know that's really interesting. Now, just to close, some of you are obviously clinicians, and you'll have people presenting with anosmia, they can't smell, or phantosmia, even worse, they can smell things that aren't there because they've had either an injury or they've had an infection which has altered their sensory neuron activity in their noses. And as I hope you've seen from the little taste thing we did with the jelly beans, it's really significant. If you lose your sense of smell, it is very, very important in terms of how it affects your life. And this also affects older people. As their sense of smell declines, then their sense of taste declines. So those of us who've got parents in institutions may sympathize that the reason why they say the food tastes of nothing may be because the food is rubbish, but it may also be because they're losing their sense of smell. And there is a charity called fifthsense.org, which is a patients group, which is very keen, especially for people who lost their sense of smell through disease or through trauma. There are ways, not of completely recovering your sense of smell, but of gradually getting back aspects of your sense of smell and beginning to experience a richer sensory world. So if any of you have patients who present with anosmia or phantosmia, then I would encourage you to put them in contact with fifthsense.org. And that's it. Thank you. Where's the stinky man smell? Where's the androstenone? Let's take it back, or people will be horrified. People come in next morning and go, what have they been doing in here? Okay, let's put the lid on it. Thank you.